Pancreatic ductal adenocarcinoma is one of the most stubborn malignancies we face. With a 5-year survival rate between 5 and 12%, it is a disease defined by its ability to resist standard treatment. For decades, the effort to fight it focused on hunting down single genetic mutations, but targeting individual genes in isolation proved insufficient because a tumor operates more like a coordinated, interconnected system than a collection of random errors. To map that system, researchers compiled a massive genomic dataset. They analyzed over 6,300 tumor samples gathered from 10 different clinical databases to identify the common threads driving the disease. Starting with an initial pool of over 15,000 altered genes, they filtered for those with the highest mutational frequency. This sifted the list down to exactly 1,617 critical genetic players. By organizing this data, scientists were able to move past the genetic chaos and reveal a structured hierarchy that explains how the disease functions at a network level. This chart shows the 5-tier classification system, categorizing 1,617 genes based on patient frequency. The red Tier 1 box contains only 6 genes. Found in nearly every study, these mutations represent the primary core initiating the cancer's growth. This lollipop plot identifies the specific locations of mutations within the most dominant Tier 1 gene, KRAS. The massive vertical spike at G12D shows its overwhelming prevalence, appearing in over 70% of all pancreatic cancer cases. Other Tier 1 genes like TP53 and SMAD4 are tumor suppressors. When they are inactivated, the cell loses its natural ability to check for DNA damage, essentially removing the brakes that prevent rapid replication. While these 6 high-frequency genes start the process, they don't work alone. To build a resistant tumor, they must interact with the hundreds of lower-frequency genes found in Tiers 2 through V. This suggests that focusing solely on Tier 1 hubs is only part of the strategy. The real challenge lies in the biological networks those hubs corrupt as the disease progresses. Researchers used cross-library pathway analysis to see how these genes interact. They found that while Tier 1 hubs are the primary drivers, they link up with hundreds of rare mutations to form highly coordinated biological modules. The data reveals 4 major functional clusters. The 2 most prominent are the blue extracellular matrix module on the left and the green neuronal signaling module in the center. There is a notable gap in the data here. Pancreatic tumors are famous for building a dense, impenetrable armor of stroma, yet the collagen and laminin genes that actually build that armor show mutation rates under 3%. The Tier 1 hubs explain why. Instead of mutating the structural genes directly, these dominant drivers act like corrupt managers, hijacking healthy signaling networks to force the surrounding cells to build the tumor's defensive walls. The neuronal network follows the same pattern. The tumor exploits natural neural pathways, using them as a structural framework for invasion into surrounding tissue. These combined mutations increase what researchers call signaling entropy, a state where the orderly communication of a cell breaks down into a chaotic plastic state. This chaotic state resists treatment. To stop it, we must target these network connections, not just isolated hubs. With this map in hand, the next step is looking at clinical practice. Do the gene tests currently available to doctors actually capture this level of network complexity? This heat map compares 4 major commercial panels. Green blocks show genes covered by Invitae, Fulgent, GeneDx, and Prevention Genetics. All successfully cover common hereditary markers like BRCA1 and BRCA2. However, Invitae and Fulgent offer 95% coverage, catching critical rare genes like TSC1 and NF1. Identifying these rare mutations is a requirement for personalized care. They give doctors the specific markers they need to navigate the complex, hijacked networks. Effectively treating a disease of this complexity requires testing that matches its scale. By detailing both the central hubs and the surrounding network, this research establishes the specific genetic targets needed for the next phase of precision oncology.